So, IGCSE Biology Paper 6, feeling a little nervous? Yeah? Don't sweat it. Over the next few minutes, we're going to completely change how you see this exam. Think of this as your ultimate toolkit for nailing every single question. And look, I get it. This paper can seem kind of daunting. It's not just about what you know, it's about what you can do. But that's exactly what we're going to work on right now. We'll break it all down piece by piece so you can walk into that exam feeling totally prepared and confident. Okay, so you ready to dive in? Here's our game plan. First, we're going to sharpen up your observation and drawing skills. Then we'll get into the nitty-gritty of data, graphs, and how to spot potential errors. After that, we tackle the big one, designing a perfect experiment from scratch. And finally, a quick checklist to make sure it all sticks. All right, first up, let's talk about what I like to call the easy marks. These observation and drawing questions, they're designed for you to score on, but you'd be surprised how many people lose marks on simple mistakes. Let's make sure you're not one of them. Okay, I know what you're thinking. These rules seem super picky, right? But there's a good reason for every single one. You draw big and with clean lines so there's no doubt about what you're seeing. And listen, the biggest one, the one everyone gets wrong, no shading. Seriously, shading is art, not science. It hides details. And always use a ruler for your label lines. It's all about being clear and precise. And when it comes to magnification, forget complicated formulas. All you need is this brilliant little I am triangle. You've got image size on top with actual size and magnification on the bottom. Just cover up what you need to find and it tells you exactly what to do. It's a fantastic trick that makes these questions pretty much impossible to get wrong. All right, so we've got the drawing part down, awesome. Now let's switch gears to observing chemical changes. There are just four key food tests you absolutely have to know like the back of your hand. Let's blitz through them. First up, the starch test. It's super simple. Add a bit of iodine. If there's starch, it's going to go from a brownish color to a really dark blue-black. You know, a fun way to remember this is potatoes. They're packed with starch. If you have some iodine solution, you could even try this at home. It's pretty cool to see. Okay, next on the list, proteins. For this, you're using Biuret reagent. If that blue solution turns purple or mauve, you've got protein. What's actually happening is the reagent is reacting with the bonds between amino acids. So you're literally seeing the building blocks of protein. How cool is that? Now for reducing sugars, and this is super important. You got to remember two things. Add Benedict's solution and heat it. Don't forget to heat it or it won't work. The best part is the final color tells you not just if there's sugar, but kind of how much sugar is there. It goes from blue to green, yellow, orange, and all the way to brick red for a high concentration. And last but not least, we've got the test for fats, the emulsion test. So why is it called that? Well, think about it. Oil and water don't mix, right? Same for fats. When you mix the sample with ethanol and then add water, the fat forms tiny little droplets, creating this cloudy, milky white emulsion. That's your positive results. Okay, so you've done your drawings, you've run your tests, you've got all this raw data. Now what? This is where we turn those numbers and observations into actual marks. It's all about perfect presentation and, this is key, thinking like a scientist about what could have gone wrong. You know, what separates a good student from a great scientist? Thinking about potential errors. The examiners love this stuff. So instead of just kind of guessing a color change, you could suggest a colorimeter for a proper reading. Instead of just counting drops, you measure the exact volume. These aren't just fixes, they're upgrades that make your experiment way more credible. All right, let's talk graphs. Honestly, if you just follow this checklist every single time, you're golden. The most important rule, the independent variable, that's the thing you change, always goes on the x-axis. The dependent variable, the thing you measure, goes on the y-axis. Just remember, the result on the y depends on what you did on the x. And please label your axes with the quantity and its units.
Okay, you hear this all the time, repeat the experiment. But why? What's the big deal? It's all about reliability. When you do it a few times, you can easily spot an anomaly. You know, that one weird result that just doesn't look right? You can ignore that outlier when you calculate your average, which makes your final conclusion way more trustworthy.
Okay, deep breath. This is the big one, the 6-mark question. This is where they're not just asking what you know. They're asking you to be a scientist and design something from scratch. But don't worry, we've got a secret weapon for this. So, let's look at a classic example. You get a hypothesis like this, and you have to plan how to test it. At first glance, this seems huge, right? Like, where do you even start? Well, that's where our simple framework comes in. We're going to dismantle it piece by piece. Say hello to your new best friend for any experiment design question, IDCSRA. Just remember, I don't care, so run away. Kind of silly, I know, but trust me, it works. It's a 6-point checklist that guarantees you'll hit every single point the examiner is looking for. Let's walk through it. So let's break it down. I is for your independent variable. That's the one thing you're changing on purpose. In this case, the volume of beetroot juice. But here's the key, don't just say change the volume. That's not enough. You have to give specific values with units, like 20, 40, 60, 80, and 100 cubic centimeters. That shows you're really thinking it through. Okay, next up, D for the dependent variable. This is what you're going to measure to see if your change had an effect. Here, it's how long the athlete can run. And just like before, get specific. How are you going to measure it? What tool are you going to use? Say it. I will measure the time in seconds using a stopwatch. Perfect. All right, now for C, and honestly, this might be the most important part, control variables. These are all the things you have to keep the same to make sure it's a fair test. Because think about it, right? If some athletes are fitter or run on a hotter day, how do you know your results are actually from the juice? You need at least 3 good ones, like age of athletes, their diet beforehand, the temperature, you get the idea. And to finish strong and grab every last mark, we have SRA. S is for a sensible safety precaution. Like for this one, maybe have medical staff present. R is for repeat. You have to say you'll repeat the experiment for each volume of juice. And A is for average. Say you'll calculate the average time for each volume. IDSRA, it's that simple. All right, we are almost there. We've covered so much, so let's just do a super quick fire round of the absolute must-knows before you walk into that exam hall. Think of this as your final briefing. This is it, your final mental checklist. I'm going to say it one more time. No shading. Label your axes with quantity and units. Name at least 3 specific control variables. Use IDCSRA for the big planning question. Always, always say you'll repeat and average, and give specific values for the thing you're changing. Get these right, and you are absolutely on your way to a fantastic grade. And that's a wrap. You've got the toolkit, you've got the strategy, you now know exactly how to think like a scientist, which is what this paper is all about. So go in there knowing you're prepared. You've got this. Now go and ace that paper. Good luck.